We are going to start talking now about the motor system. And in this first part for Chapter 13 and 14 are much like Chapter 10 and 11, where we talked about the somatosensory system, peripheral and central. Chapter 13 is the peripheral part of the motor system, and Chapter 14 is the central part of the motor system. So we're going to start with a review of muscle function and then move into motor neurons and spinal motor function. So the learning objectives for this part of the lecture is we're going to talk about muscles and muscle tone. What is muscle tone and how is it assessed? And where does the decision about motor activities get made? Why is it important for a sarcomere to be at optimal length? We've talked about sarcomeres in previous classes. It's the smallest unit of muscle function. And what happens to a muscle if a limb is immobilized with the muscle in a shortened position? So let's find out. So here's our milepost for where we are with regard to our association to the cerebral cortex. So we had all that on the left side of the graph, we had all that information coming in from the somatosensory system going to the primary sensory cortex, which discriminates the intensity and quality of sensations. And then it goes to the secondary sensory cortex, where we are involved in the recognition of sensation. Like, what is that? I know the intensity and the quality. Now let's line it up with what we know, figure out what it is. The association cortex is a huge part of our brain with goal selection, planning, monitoring, interpretation of sensation, emotions and memory and processing. And if the association cortex takes all the information from the somatosensory system and decides that we need to have a motor act, it sends that information, that processed information, to the motor planning areas. And in the motor planning areas, we get movement composition and sequencing, and then it goes to the primary motor cortex, and the cell bodies of the upper motor neurons, or the motor tracts, are in the cerebral cortex, in the primary motor cortex. And that's where we get cortical motor output that goes to the spinal cord and the lower motor neurons, which those are the motor neurons that we think about when we're talking about peripheral nerves. This all has to happen before we get that output that is our efferent nervous system output from the cortex to the muscles. So that's where we are in this whole big picture. So neural activity in the motor system begins with a decision being made in the anterior part of the frontal lobe. So the anterior part of our frontal lobe is our decision-making place. That's the place that where we decide what actions we're going to take. That information is then sent to the motor planning areas and some control circuits, which we'll talk about a little bit later in chapters 15 and 16. And the control circuits are in the cerebellum and the basal ganglia, and they regulate the activity in the motor tract neuron. So the brain says, yeah, we got to do this, and the cerebellum and basal ganglia say, wait a minute, let's do it under control. So where there's lots of input going into a motor act, but the initial decision is made in the anterior part of the frontal lobe. So the motor tract neuron, also known as the upper motor neuron, delivers signals to spinal interneurons and to the motor neurons, also known as the lower motor neurons. So the lower motor neurons go from the spine to the muscle, and they transmit signals directly to the skeletal muscles, eliciting the contraction of muscle fibers that moves the upper limbs and fingers. Yay, right? That's what we wanted to do. Voluntary movement is controlled from the top down. So it goes from the brain to the spinal cord to the muscle. We go from that anterior frontal lobe to the spinal interneurons to the lower motor neurons, which take it out to the neuromuscular junction. So there's no bottom-up control on the muscles. It's all top-down control. So just a little bit of a review of skeletal muscle structure and function. Skeletal muscle is excitable, contractile, extensile, and elastic. And all of those characteristics of skeletal muscle are important to its function. So each individual muscle fiber consists of myofibrils arranged parallel to the long axis of the muscle fiber. And those myofibrils are made up of individual sarcomeres. Sarcomeres are the base unit of muscle. So within the sarcomere, the protein titin, which is the little orange line on this, and on the next slide I have a bigger picture of this, we'll look at it. But titin maintains the position of myosin relative to the actin and prevents the sarcomere from being pulled apart. So the titin is a structural protein, and it is largely responsible for the tension or tone in the muscle, or it's one of the big factors in resting muscle tone. So here's that bigger picture. In the top, letter A, the titin is stretched, it's tightened, it's preventing the sarcomere from being pulled apart. So we have that actin-myosin crossover that's so important for a good muscle contraction. In the bottom picture, the titin is in a less stretched position, and it's maintaining the myosin in the center of the sarcomere. So we're trying to maintain that actin-myosin crossover. I hope you remember that from kinesiology and anatomy and physiology, the role of actin and myosin in the contraction of the muscle. But titin is that structural protein that's helping maintain the structure of the sarcomere and the tension of the muscle to some extent. So the muscle contraction is produced when the actin slides relative to the myosin. Repeated attachment and swiveling and detachment of myosin heads produce the contraction of the muscle. So hopefully that is a review, and you're saying, oh, right, sliding filament theory, I know all this stuff, right? So when we talk about resistance to muscle stretch, muscles behave somewhat like springs. In other words, if you took a spring and you stretched it all the way out, it would be harder to stretch it further than stretching the same spring when it's shortened. So if you ever do Pilates reformer, the spring stuff totally makes sense. You're like, oh yeah, I'm stretching springs all the time on the reformer. But the resistance to stretch in muscles depends on their length. So a stretched muscle is going to have more resistance to stretch, and a shortened muscle is going to have less resistance to stretch. Active muscle contraction, that structural protein titin, and weak actin-myosin bonds determine the total resistance to muscle stretch. So the actin and myosin, when they're just sitting there, when the muscle's at rest, you get some weak bonds, and it's sort of like old Velcro, if you want to think of it that way. And then when you start to move, you have to break those bonds, and that resists the muscle stretch a little bit. Active muscle contraction resists the stretch, and then the structure of the titin also resists the stretch. So muscle tone is a term we're going to talk about. So part of the time in some of the different classes in this program, you have to relearn some things that we've talked about, some terminology that's used in other areas. So a lot of times in the fitness world, when you talk about muscle tone, you're talking about, like how firm and shaped are your muscles. But when we're talking about muscle tone in a physical therapy and neurological setting, muscle tone is defined as the resistance to stretch in a resting muscle. Clinically, we use passive range of motion to assess muscle tone. So you've had a chance to do some passive range of motion and some muscle testing with your classmates in lab, and you know that someone who doesn't have an injury and there has normal muscle tone, when you do passive range of motion, there isn't a lot of resistance. There's minimal resistance to passive stretch. So that's with normal muscle tone. You can move someone through their range of motion pretty easily, a passive range of motion. The normal resting muscle tone is provided by that structural protein titin and those weak actin myosin bonds. So when we're testing for muscle tone in the clinic, we're going to do passive range of motion, and normal muscle tone means you can move that person through their passive range of motion with very little resistance. Okay, if someone does have resistance, we'll talk about abnormal muscle tone a couple lectures forward. So when a healthy innervated muscle, it's important, is continuously immobilized in a shortened position for a prolonged period, remember we're trying to maintain that length tension relationship in the muscle, and so the body will adapt by removing sarcomeres from the ends of myofibrils. The loss of sarcomeres is a structural adaptation to the shortened position, so the muscle maintains its length tension relationship and generates optimal force at the new resting length. Conversely, when a structurally shortened muscle is stretched, it'll quickly reach the limits of its elasticity and it'll be resistant to stretching. So the muscle is actually shorter. And I don't know if you've personally experienced when you maybe you're doing a new strengthening program and you haven't taken the time to stretch a lot and your muscles are shortened. You have a lot of resistance to stretch. The muscle's resistant to stretch. But really, we're actually talking about a contracture here where the muscle is continuously immobilized. It's not just a new strengthening program, but you have, like, say that you have your elbow flexed at 90 degrees for a prolonged period, meaning over days / weeks / months, you will lose sarcomeres. The muscle will actually shorten. So if the muscle is immobilized in a lengthened position, the muscle will add new sarcomeres. So chronic loading of muscle, as with physical training, stimulates mechanoreceptors that switch on genes for producing collagen and extracellular matrix to improve the muscle's tensile strength. So physical training is good. We know that, but this is why physical training is good. So if you immobilize the muscle in a shortened position, you will lose sarcomeres. If you immobilize the muscle in a lengthened position, you will add new sarcomeres. So the immobilizing the muscle in a lengthened position is the basis of doing dynamic splinting, where you have someone with a contracture you're trying to lengthen. You put them in a splint for several hours a day, so immobilized in a lengthened position, to encourage the muscle to add new sarcomeres. So you can actually change the length of your myofibrils by how the muscle is in its resting position. So besides muscle resistance to movement, we also have joint resistant to movement. Cocontraction refers to the simultaneous contraction of antagonist muscles. So the agonist and the antagonist contract at the same time, and we do this to stabilize joints. A lot of times we use co-contraction when we're trying to do precise movements in the upper limbs. Like say that you're engraving on wood, and so you're contracting all your muscles to stabilize that hand so you can do your engraving. In the lower limbs, it allows you to stand on an unstable surface. So say that you're on the bus and everybody else is sitting down and there are no seats left, you have to stand. And so you co-contract your muscles to stabilize your joints and allow you to stand on that unstable surface. That's why you should give old people your seat on the bus, because it's harder for them to do co-contraction. So if somebody older than you is standing on the bus and you're sitting, you should give them your seat.